Hello dear students, here I am going to discuss the microscopic study of Plasmodium vivax. As you know, there are different species of Plasmodium which cause the disease malaria. Plasmodium vivax is a protozoan parasite and a human pathogen. This parasite is the most frequent and widely distributed cause of recurring malaria. Plasmodium vivax malaria infection can lead to severe disease and death, often due to splenomegaly. So, I will discuss here how to diagnose the Plasmodium vivax in the RBCs of human samples. For studying permanent slides of different parasites or specimens, first of all, you have to keep a permanent slide under the microscope, and then you need to recognize special features of that particular parasite in order to recognize. So before I will show you the actual image of this parasite, that is Plasmodium vivax, which we observe under the microscope. I want to show you the general image of this parasite when it is under the red blood cell. So here you have a red blood cell. So under this red blood cell, there is another spherical structure which has special features. For example, in this case, you have one or more red chromatin dots, and then you have blue cytoplasm. So this image or cartoon shows you how to recognize parasite, though you will not find this type of structure under the microscope, but this particular image gives you an idea how to recognize the parasite. As you are aware that malaria parasite undergoes two major stages when it enters into the bloodstream. One stage is known as pre-erythrocytic stage and another is known as erythrocytic stage. Under erythrocytic stage, there occurs another four substages, and these four substages can be recognized by spatial morphological features of this parasite. So these different four stages are recognized as ring stage, trophocyte stage, scion stage, and gametocyte stage. For example, when you have a permanent slide of parasite having ring stage, you can recognize it by having a red dot like structure under the microscope. So when you have a trophocyte stage, a slide of trophocyte stage, you can recognize it by showing proliferation, and the blue cytoplasm is evident under the microscope. And when you have a slide having scion stage, you can find numerous red chromatin and also brown pigments on the surface of this parasite. And finally, when you have a slide having gametocyte stage, it shows numerous brown structures and a single red dot-like structures or red chromatin. So the crux here is to tell you that these different stages can be recognized by spatial features. Here in this slide, you can see the actual or real images of malarial parasite when we observe it under the microscope. So on the top, you can see the ring form where student can recognize this ring form easily by observing ring-like structure inside the RBC. And then you have trophocyte stage where you can see the haphazard movement of this parasite inside the RBCs. And then starts the scion stage where you can recognize small dot-like structures and the growth of this malarial parasite inside the RBCs. And finally, you have gametocyte stage, gametocytes, emerging from this parasite, and you can see it shows more proliferation and sort of growth in the RBCs. So this is the magnified image of earlier slide, where you can see the magnified images of different stages which you observe under the microscope. You have a ring stage where you can recognize ring-like structure. Then you have trophozoite stage where growth of this parasite has occurred and has shown proliferation. Here at the next stage, Scission stage, where you can see small dot-like structures, because this parasite is now showing proliferation at the highest level. And then finally you have gametocyte stage where full proliferation has occurred. So these are the real images which you can see under the microscope. As you have already observed, malaria parasite under the microscope, after the completion of microscopic study, you need to draw the diagrams of different stages. So here in this slide, you can observe the drawings of different parasites, which is based on your observation. In this slide, you can see the morphological differences of different stages of Plasmodium parasites when observed inside the erythrocytes. So you can observe morphological differences at trophozoite stage, at Scission stage, and at gametocyte stage. So by this way, you can be able to recognize the particular feature of a particular parasite. By that way, you can then recognize the specific parasite, and in this way you will be able to identify and recognize particular slide which is given to you during examination. I hope you have understood this practical, and now only thing you need to do is to observe permanent slide under the microscope. When you are back to college, you can do that activity. Thank you.